Welcome to our school, JMB. Let's begin with the chapter number 9, standard 11, excretory products and their elimination. By various metabolic activities, animal produce metabolic waste: ammonia, urea, uric acid, CO2, water, Na+, K+, Cl-, phosphate, sulfate, and so many other things. All these should be partially or totally removed from the body. Ammonia is highly toxic, require large water. Urea is moderate toxic, so required quite less water than the ammonia. Uric acid is least toxic. All among these three, ammonia, urea and uric acid, are the nitrogenous waste. So it should be completely removed from the body as early as the possible. So this is the category. Ammonia is higher toxic, less is the urea and least is the uric acid. Right, type of the animal according to the excretion is the aminotalic animals, ureotalic animals and uricotalic animals. Let's begin with one by one. Let's talk with about the aminotalic first. It's excretion of the ammonia. Certain amphibians and aquatic insects do this. They excrete ammonia, so they are called as the aminotalic. Examples are important for the MCQ. It is highly soluble in water, so by the diffusion process it can easily removed through the help of the body surface from higher concentration to lower concentration. Mostly fish with the help of the gills do that. Right? Kidney does not play any role in this mechanism. It's easy. Then ureotalic is the excretion of the urea. Right? Mammals, amphibians and marine fishes will do that. Okay, see, I am going to show you how urea is excreted. It's very nice mechanism. I make a flow chart for you. See, yes, urea, it's a nitrogenous waste, right? Normally, by all metabolic activities in body, ammonia is produced. Right, this ammonia is transported to the liver with the help of the blood. In liver, ammonia is converted to urea. Urea then transported to kidneys. Now in kidney, urea divided into two parts. A fraction of the urea, small amount of the urea, remain in the kidney matrix. For what? Osmoregulation. Right, for pH balance. And remaining other urea is excreted through the urine, right, by the kidneys. So here major role is played by the kidneys. So this is what ureotalic animals do, excrete the urea. It's a nitrogenous waste. Normally by the product ammonia is the produced only by the metabolic activities. It is transported to the liver. Liver converts ammonia to urea. Fraction of the urea is remain in the kidney and other urea is excreted. Am I clear to you? It is very important. Then last is the uricotalic animals. Uricotalic excrete the uric acid. Examples are reptile, birds, snail and insects. It is less toxic, right, and obviously less soluble in water also. So it is normally excreted in form of the pellet or paste like Colgate paste. Energy requirement is also very less over here. So see, what is the relationship of a water and energy requirement for the excretion? It is inversely proportional. If water requirement is large, then energy requirement is less. Consider the aminotelic. Aminotelic animal required large amount of the water, so very less energy is required, right? Then ureotelic require moderate water, obviously moderate energy. This is the energy requirement. Okay, and uricotelic animal requires large amount of the very less amount of the water, so large amount of the energy requirement. See, for the water requirement, aminotelic large water, uricotelic less water, and uricotelic is least water. And if we talk about the energy requirement, then yes, opposite, urico required the large energy, then ureo is moderate, and then aminotelic required very less energy, right? In standard 11, in chapter number 4, we already studied the organ of the excretion. So these are the excretory organs. Protonephridias and phlegm cells are there in the platyhelminthes and rotifers and cephalochordates, particularly amphioxus, and some of the annelids. Nephridias are there in the annelids and the earthworm. Malpighian tubules are there in the insect, almost in or mostly arthropodas, and it is there in the cockroach. Green glands are there in crustaceans and in the prone, and kidneys are there in almost all vertebrates. So let's begin with human excretory system. Consists of pair of kidneys, a pair of urinary bladder, a pair of ureter, urinary bladder, and the urethra. Kidney is actually bean shaped organ, right? It is located between the last thoracic and third lumbar vertebrate. Length is about 10 to 12 centimeter, width is about 5 to 7 centimeter, thickness is about 2 to 3 centimeter. Weight is about almost 120 to 170 gram, right? The inner surface is concave and outer surface is convex. It has two regions, the outer region and the inner region. Outer region is cortex and inner region is medulla. It has a tuft capsule surrounds it. Now see, if this is a kidney, there is a notch like structure. This notch like structure is called as the hilum, right? In this, a funnel shaped structure is there called as the renal pelvis. This is the renal pelvis. Pelvis forms a conical projection like this. These conical projections are called as the calyx or calycis. Above this projection, a masses of the cell are located like this. That is called as the renal pyramid. This is called as the renal pyramid, right? Now the cortex is extended between the medullary pyramids. See this is the cortex region. It is extended in between the medullary pyramids like this, like over here, like over here, like over here. That is called as the column of Bertini. It is called as column of Bertini. Hilum is the region in which artery and nerves enters. artery plus nerves enter, and vein and the ureter leaves. Done. So this is the general information about the kidney. This is the diagram which is important. You have to explain this with a diagram, and it is of the 3 marks question. Okay, let's begin with the microstructure. What we see inside the kidney. Inside the kidney we can find millions of the nephrons. Millions in each kidney. Millions of the nephrons. You can say it is the structural unit. It is the structural unit. How nephrons are formed? It is formed by the glomerulus and renal tubule. Right? 2 things are there, by the glomerulus and renal tubule. This is a Bowman's capsule. Inside the Bowman's capsule, a renal artery enters. That is called as the afferent artery. And the leaves, that is called as the efferent artery. And between the afferent and efferent there is a network of capillaries. Whole this is called as the Malpighian tubules. Yes, this is the glomerulus. A network of capillary inside the Bowman's capsule is glomerulus, and this is the Bowman's capsule. Combinedly called as the Malpighian tubules. So this is what the structural and functional unit is there, right? It is converted to the small neck-like structure. Then it is formed a highly coiled-like structure called as the proximal convoluted tubules. Then it converted to the U-shaped hairpin-like structure, that's the loop of Henle. It is called as the loop of Henle. This is the descending part, means it moving downwards. This is the ascending part, it is moving upward. Then it is again converted to the coiled structure, that is the distal convoluted tubule. And now lastly it enters into the collecting duct. So this is what whole a nephron look like, right? Structural unit which do the main function of the filtration. Am I clear to you? Now this afferent arteriole comes out from the Bowman, and it forms a network of a capillary around the loop of Henle. See, like this. This is called as the vasa recta. Here I have made a small flowchart for you. See this Bowman's capsule. It is a double-walled cup-like structure enclosed in a glomerulus. Bowman's capsule and glomerulus combinedly form Malpighian body, or you can say renal corpuscles. Then then tubular structure it is converted, called as the proximal convoluted tubules. And then U-shaped hairpin-like structure, that's the Henle's loop. It has 2 parts, descending part of the Henle and ascending part of the Henle. Then it is converted to the highly coiled structure, distal convoluted tubules. Then it opens into the collecting duct, and then it is converted to the ureter. Clear? So this is what the diagram of the textbook. See, this is the loop of Henle, and this afferent arteriole forms a network around it called as the vasa recta. Vasa recta is important. We have to study more things about the vasa recta in this. So see this is the vasa recta. Then 2 type of the nephrons we have, that is the cortical and the juxta. In cortical, the loop of Henle is highly reduced, very small, whereas in juxta it is well developed, so it runs deep inside the medulla. See, vasa recta is the network of the capillary around the loop of Henle. Here Henle is not there, so vasa recta is not formed. Instead that capillaries forms a network around the renal tubule called as the peritubular capillary. It is about 85%. Loop of Henle is well developed over here, so runs deep inside the medulla, so vasa recta is present, yes, and it is almost 50%. You may ask in the difference, very important question for 2 marks, explain the difference of cortical and juxta nephrons. Clear? So here loop of Henle is reduced, here well developed, vasa recta is absent, vasa recta is present. Capillary form a network around the renal tubule called as the peritubular capillaries. Here peritubular capillaries are not present. It is about 85% and it is about the 15%. Am I clear to you? Okay. Thank you. See you in the next lecture.